Next, it is my pleasure to present our next speaker. She is a MacArthur Fellow and McKnight Distinguished Professor in Entomology at the University of Minnesota. Her research efforts focus on protecting the health of all bees, breeding bees for their natural defenses against diseases and parasites, promoting sustainable beekeeping practices, and propagating floral-rich and pesticide-free landscapes to support nutrition, health, and diversity of bee pollinators. I'd like to present Marla Spivak.
Hi, good morning. I had a great night, it's great weather. I don't want to go home. Can you hear me? This is all right. Do you want me to speak louder? Okay. A couple of funny things happened yesterday during my talk. I was talking about how I began beekeeping here with the Cole family in Bosque Farms. Rick Cole, the oldest son, was in the audience, and I didn't know it. He came up to me afterwards. I didn't say anything bad about them. They were wonderful people, wonderful mentors. It was great to see Ricky, and he was 14 when he was the oldest kid out of 5. I was 19 at the time, and we were all like one big family. It was great to see him. And the other funny thing is that I was kind of teasing Megan Mahoney, and Megan's dad was here. Is he here today?
Yeah.
Oh, good. I think the world of Megan, and I like to tease, and I hope I said nothing bad against her. Megan's amazing.
I like her.
Yeah. Okay. So today's talk, thanks, is a research talk, and we have lots of different research projects going on, and I'm going to breeze through many of them, focusing a little in depth on a few and breezing through others, and hopefully I'll have lots of times for questions. So again, this is our website, and there is Megan featured on the website because she had the absolute best bee beard of anybody, and that's the truth. And here's all of the people in my lab. And it is not a requirement that people do a bearded bees to work in the lab, but for some reason they come. We always do one for our big state fair we have in the Twin Cities, and we do a practice day for anybody that would like to do it. We do it at the lab, and so we put a bearded bees on somebody, and then we take it off and put it on the next person, and we've passed this beard on to about 7 different people before the bees said enough, and I'm not doing this anymore. But these are some of the wonderful people in my lab. So the main research focus that I've done for years and that we do is promoting bees' own natural defenses. And as I was saying yesterday, bees have their own healthcare system, and in research language we call it social immunity. And so an individual bee has an immune system. They have very good immune systems. The difference between a bee immune system and our immune system is that bees and all other invertebrates, all insects and everything without a backbone, does not produce antibodies. So their immune system doesn't have that long-term memory, so you really can't give them a vaccine for them to produce antibodies, okay? But otherwise, the rest of the immune system is there. It's a very general immune system where the cells are able to detect foreign microbes, and they encapsulate them, they break them down, they have proteins that break down foreign microbes, etc., etc., etc. The same one we do, but they don't have the antibodies. But when a group of bees get together, then they each have, it's 50,000 bees in a colony, they each have their own immune systems, and then each bee in the colony starts acting analogously to a cell within the whole body. So if the colony is the organism, if it's the animal, then each individual bee is like a cell within that animal. And some bees start doing things that are very similar to what our immune system does within us. So some bees, for example, are very, very good at detecting foreign microbes, and these are called hygienic bees. And they detect when a larval or a pupal bee is infected with a disease or it's infested with a mite. They detect it through odors, and they go in and instead of breaking it down, they actually throw it out of the nest. okay? They discard the bee out of the nest if it's sick, okay? That's very similar to how an immune system works. So that is what hygienic behavior is, and this diagram shows a bee that's detected a pupa that has mites in it, and the bee opens up the cell and removes the whole pupa. And the mite that's on the pupa, sometimes the mother mite can escape. But what happens is if the hygienic bees keep interrupting the mite's reproductive cycle, because she's in there laying babies, right? She's laying eggs. If the bees keep interrupting the reproductive cycle of the mite, eventually that mother mite runs out of sperm. Or she goes into the next cell, she's interrupted here, she crawls out, she goes into the next cell, and her pattern, the mites are really rigid in their behavior, and she's not ready to lay a male egg, for example. So she only lays females, and then there's nobody to mate with. So the constant disturbance of the reproductive cycle messes up the mites and reduces the mite load. So we've bred these bees for a very long time, but more currently, we've been moving into another social immunity behavior, and that is propolis collection. So bees collect these resins from trees, and in our area they're mostly poplar trees, and I'll show you how we found that out. So, well, actually not poplar, they're in the genus Propolis, which contains poplar and cottonwood and aspen. Okay, so those are the main sources of resin for the trees in Minnesota, in our area. And down here, it may be things like rabbit brush, some cottonwood for sure in your riparian areas, and not sure what else. You think it's juniper? Okay, you guys tell me then, that's great. So these resins, okay, this picture on the right is a bee in Brazil actually, and she's scraping a very green resin off the leaves of a plant, Baccharis plant, and she's going to stick those resins on her back leg. Hold on, sorry. Here we go. She's going to stick those resins on her back leg just like pollen. But they don't, and they carry the resins back to the nest, and they use them in the nest cavity like glue. They do not eat these resins. Once the resins are inside the colony, we call it propolis, or propolis. Tomato, tomato, it doesn't matter how you say it, okay? So the resin equals propolis, and inside a bee colony, the bees may add some wax to it, the resin, but they, as far as we know, they are not modifying the chemistry of the resins at all. It comes in as resin, it stays as resin. The plants secrete these resins because they protect the plant. They protect the plant leaf buds from infection, and they prevent the plant from UV radiation, and they prevent it from herbivores, other leaf-eating pests, from eating the leaves of the plant. And the bees are very good at cueing in on plants that secrete these resins, and by odor, probably, you know how propolis smells, it's wonderful, I think. And they go scrape these resins off, carry them on their back legs, take them home. Now, it's really hard for bees to collect resin. So I'm going to use resin and propolis throughout this talk interchangeably, but you'll know what I'm saying now, right? If I go back and forth, okay. I try to say resin from when they're outside and propolis when it's inside the nest, but I get goofed up sometimes. Okay, so it's very hard for the bees to collect the resins because once the bee gets, they're very, very sticky, as you know. It's probably all over your clothes, right? It's all over everything I own. And then once they get into the nest, the bees have a really hard time getting the resin off of their back legs. They cannot get it off themselves. Another bee has to help them pull it off of their leg. It's like getting a wax, I think. Okay, so these resins are hard to collect. The bees are not eating them, and I want you to remember that because I give this talk to beekeeping groups and they want to feed their bees propolis. It's an extremely powerful antibiotic. Please don't feed your bees propolis. We don't think that's good. They don't eat it themselves, okay? But we do know from our prior research that bees, if they're given a propolis envelope, and this was work that Tom Seeley showed before I get to this particular slide. Tom Seeley, and you know him, he came here, I guess, opened up a bunch of wild bee nests that were in tree cavities for his PhD work. and described the inside lining of these tree cavities, and it was just solid propolis. Is that if we were a beehive, we were bees in this colony, we would be painting the walls here, every crack and crevice, and varnishing the walls with propolis in our hive. That's how it would look in a natural bee cavity. But in a Langstroth hive and in a top bar hive, even, they don't put it on the walls like they do naturally. And the reason is that we use smooth lumber, because the inside of a tree cavity is very, very rough. And that encourages them, they don't like those cracks and crevices, that encourages them to put that varnish layer of propolis inside. And so we know from this guy's research that propolis, when they have that whole envelope in the colony, it helps the bee's immune system, because these are such powerful antimicrobial compounds in the resins that it's fighting off microbes within the nest cavity. And so then if there's fewer microbes in the nest cavity, the bee's immune systems don't have to be activated. When your immune system is activated, when yours or a bee's or anybody's immune system is activated, it's at a cost. For bees, they're either going to produce less honey, they're not going to live as long, they're going to wear out faster. Immune system activation is tough. So anything that keeps the immune system quiet is a good thing. So the questions we're asking, do bees with a propolis envelope have less disease? Where are the resins coming from? And are some better medicines, if you will, than others, some more antimicrobial? And do bees choose medicine? Do they self-medicate? Are they choosing the resins? So these are the questions we're trying to get at right now. So here's the first study with Mike Simone Finstrom, PhD student, and what he did was to make a propolis extract. So you take propolis, you dissolve it in 70% ethanol, and you just paint it on like varnish. And we use these small nuke boxes. So the one on the left has nothing, just the ethanol as a control, and the one in the middle has the green propolis from Brazil, known to be very, very high in antimicrobial properties, and the one on the right, the reddish one, is stuff from Minnesota. Propolis collected from colonies in Minnesota, extracted, painted on, put bees in there, paint marked the bees, collected them when they were 7 days, and measured their immune systems. And that's how we found out that it's really helping their immune system. The immune system activation was lower. Okay, so here's Renata Orba, a new student. Well, she's going to be finishing this year. But in a new set of studies, she wanted to know, rather than painting on the propolis, is it better if the bees do their own propolis envelope? This photo here is inside of a tree cavity looking out. So this is the bees looking out their front door. And look at all of this propolis. All of this orange, yellow, green, brown stuff is propolis, a pretty thick layer, especially around the front entrance, but all the way through the nest. So her question was, should we give them a whole propolis envelope, let them do it naturally, or is it enough just to throw a propolis trap on top of the colony? You've seen these commercial traps. They have these 16-inch slits. And bees are so fastidious, they don't like those little cracks. They'll fill it up with resin. If it's a wider crack, they fill it with wax. Smaller cracks, they fill with resin. So she set up a bunch of colonies, 2 different times, 2 different years, and in some of them she just she cut apart all very expensive propolis straps. It's all right. She stapled them inside the colonies like this. Okay, so she had colonies that had, in this way they would stick it through the propolis trap and make a natural envelope. And then in other sets she put a propolis trap on the top, and others she just left them as controls. And this is what it looked like if you took the propolis traps off. We kept them on, but that's what it looked like. So that's kind of the natural, artificial kind of, naturally induced propolis envelope, let's put it that way. Okay, and then here's data showing their immune systems. And you don't need to there's no quiz in this thing, but here's 3 different proteins that are produced by the immune system, and these are antimicrobial proteins. They recognize microbes, break them down. And the white bars are the control colonies with no propolis. You can see their immune systems were elevated. And you can see the brown ones are the colonies with propolis envelopes in them, and their immune systems were quieter. And the ones with the propolis traps were kind of variable and intermediate. And Interestingly, if you follow those same colonies all the way through our pretty severe Minnesota winter and into the next spring, what starts happening is no differences now. So we're stuck on this problem. We repeated this twice, saw it twice, where by spring there was no difference. I think the propolis, excuse me, is losing its activity, and that bees need to bring in new fresh stuff. So this is something we easily tested. We have a collection of propolis from last fall, 2014, where we know its microbial activity, and I'll show you how we know that. And then we'll go out early this spring and collect it from the colonies to see if it's in May. I mean, that's early for us. And collect the propolis to see if it's comparable in activity or if with time it loses it. I have a feeling it's just not there, and by June when it's warming up and bees are bringing in more propolis, it would remain active, or it would gain activity again. This is the same. Okay. So that's where we're at with that. It's looking like the propolis envelope, the natural one, is really the best thing for bees. Actually, the propolis trap on the colony, I would say, is not good for bees, especially in northern climates, because bees need to evaporate moisture. Moisture needs to whip up out of the colony in the middle of winter. And putting a moisture barrier, a trap on the top, is not good. Their virus levels went way up in those propolis trap on the top colonies, so don't leave those on for a minute. Yes?
Have you looked at how rough the wood would need to be to
Yeah,
serve the same purpose?
let me get to that. I have a photo. The next question that we asked was, all we know so far is it's good for their immune systems, but can it actually help them with some of their diseases? So that first student, Mike Simone, put propolis, in his case he painted the propolis extract in some colonies, and then he challenged colonies by infecting them with chalk brood, which is very easy to do if you make up a pollen patty and you put a few chalk brood mummies in it. So if you collect your own pollen and you make your own pollen patties, make sure you don't put chalk brood in those pollen patties. Don't collect any chalk brood mummies that fall into your traps. Anyway, he put some pollen, chalk brood mummies homogenized in the pollen, fed it to colonies, and then measured how much infection they had. And colonies with a propolis envelope, this is how many chalk brood mummies they had on average, about 15, and this was about 6 colonies on average. And this was with that no envelope, 108 mummies. So really significantly reducing chalk brood infection in the colonies. So Renata came along and said, okay, what about American foul brood? Because in the laboratory, propolis is very active against American foul brood. So she again set up her colonies with these envelopes and controls, and in this time, instead of feeding them in pollen, to inoculate or challenge a colony with American foul brood, you make a sugar solution with spores and you spray it on the combs. And that gives them American foul brood. And what she found is after two months after infection, and I'll explain this graph to you, they had less American foul brood. And so the way this works is, if you've seen a colony with American foul brood, some of the bees uncap the cells. You can see that brown gooey stuff in there. And that's clinical symptoms of American foul brood, a bacterial disease. But sometimes the bees don't uncap all the cells that contain American foul brood. So what she did was take everything back in the laboratory at the end of the experiment and open up every single cell remaining in these are small 5-frame nukes, every single cell remaining in there. And those that were already unsealed, here's how much American foul brood, 20 cells per frame in the controls and in the propolis envelope colonies, and cells that were still sealed, not opened yet by the bees, had this much infection versus this. Okay? So significant reduction in American foul brood, which is really encouraging. All right, so where are these resins coming from? And for this, I had to collaborate with some plant chemists who do this fancy thing called metabolic fingerprinting, where you run samples of resins through liquid chromatography mass spectrometry, can't even say it, piece of equipment, and it gives you a fingerprint of all the compounds or all the metabolites in that resin. And so Here's some fingerprints of cottonwood and balsam poplar and a poplar hybrid and a pine, for example. Okay? And then what you do is you tell your graduate student to climb a bunch of trees around campus and collect resin from a bunch of these different trees and bring in a pure resin sample directly from the tree, and then sit in front of a colony and collect bees returning with resin. And you take each of those and run them through your LCMS and compare their fingerprints. And this is what we did. And now this is how we know where the resin's coming from. Is that here's the leaf fingerprint, here's the bee fingerprint, and we know it's cottonwood. Not easy, okay? So here's the trees that a different student, Mike Wilson, climbed. And he's a chemist, so it was fun to see somebody, a chemist, outside climbing trees. 5 pine, 3 spruce, 3 fir, 2 populus, that genus, horse chestnut. And we found that bees were predominantly collecting cottonwood. And it wasn't the most predominant tree around campus, but it's what the bees were flying to collect. And there's Mike Wilson. So now with these resins, he can do a test to see whether some are more antimicrobial than others. And generally the way you do this, maybe you remember if you took microbiology, is you you have an agar plate with a medium, food medium in it, and you put bacteria on the medium and they start growing, right? And then what you can do is take a little filtered paper and you put an antibiotic on it. In this case, this is tylosin. And this zone here, this clear zone, tells you how much it inhibits the growth of the bacteria on the plate. Here's the bacteria, here's the antibiotic, and that's the clear zone, the zone of inhibition where the bacteria cannot grow. You just measure that. So here's an example of propolis as opposed to tylosin. It's pretty good. Tylosin is a specific antibiotic. This was just propolis. We don't even know how much antimicrobial property it had. But this takes a fairly long time, so we used a very, what we call high throughput, rapid way to do it. You just culture the bacteria in a liquid medium. You run it through a spectrophotometer, which can measure the density of the medium. So if the antibiotic's working, the solution becomes clear. And if it's cloudy, it's full of bacteria, it's cloudy. You can just measure that. So what we found, this is interesting, here is cottonwood, and this is, the higher up on the list, is the more antimicrobial it is, okay? So cottonwood isn't the most antimicrobial. This is balsam poplar, much better. This is, I think, white spruce. And it is really, really antimicrobial, but the bees were not collecting it so much. Now this was testing their activity against this bacterium, which is the bacteria that causes American foulbrood. So this is just one bacterium. We tested again with fungi, the chalkroot fungus, and kind of found the same results. Okay, this is what we're at with this. We don't know yet. We do know that if you make a colony sick with chalkroot, the colony brings in more resin afterwards. So we do know that they're kind of self-medicating, but we don't know if they're changing their medication, if you will. If they're sick, if they're bringing in, if they go search out white spruce, for example. We don't know that yet, but that's where the research is going. It's pretty exciting, so you have to stay tuned. So the direct application, and this gets to your question, can we just roughen up the insides of a colony? And this we just used, what did Gary, my technician, use? I forget what a rasper like. And this is what we got. It's not as thick as they would do naturally. Here's propolis in the frame rests where everybody hates it, right? Where it sticks your frames down. And when not shown here, but between the boxes. So if you just simply roughen it up a little bit, I don't think it gives you the amount of propolis envelope that they would do naturally. How much is enough? I don't know. Anything is better than what we're giving them now. I do know that. Okay, yes?
Is it possible that if when you go in the hive, if you put it on a table saw, say cut small seeds on the inside?
Yeah. So in Brazil, they have, the Africanized bees bring in so much propolis. It's a genetic trait, and those bees, incredible propolis collectors. And they can just take whole slats out of the whole side of the box, like a half-inch slat, and the bees will fill it all in with propolis. Our bees won't do that. So I think if you did put in slits like that, I think they would fill them, definitely would fill them 16-inch slits, a lot of them, yeah. That would be a great way to do it. Yeah.
I was just going to ask that, like with our mating nukes, we make grooves, the standard box, we make grooves in it to slip in on
Yes.
these night dividers. And guaranteed, yeah, they'll fill those little
Yeah.
slits. But those are vertical instead of horizontal. Does that
Right.
make a difference?
No. No. Okay, now I'm going to move a little bit quicker. This is another student's project. This is Mike Gobler. He finished his PhD. He developed the very first honeybee cell line. And let me explain what that means. Have any of you read the book, The Immortal Life of Henrietta Lacks? If you haven't read this book, you should go read the book. It's how they developed the very first human cell line. And what this is cells from the human body, or tissues from the human body that are cultured in a laboratory, and that you're able to pass from one medium to another, and you just continue the growth of these cells indefinitely. And with those cells, you can infect them with what we call intracellular pathogens, those diseases that only live inside of a cell, like a virus, like eczema. They only can live inside of a cell. And so the only way to really study what they're doing is to get them inside cells. And then now you have cells within a bee, bees within a colony, colonies in an apiary. It gets really complicated if you try to study viruses and eczema at a colony level. So with the cell line, for the first time now, we can look at how viruses work within the cell of a honeybee. We can look at all kinds of questions about genomics, proteomics, all kinds of questions about what's going on at the cellular level. We can come up with treatments. For example, does propolis help with honeybee viruses? This is something we can study with the cell line, okay, in the laboratory. And once we have this information, then we can extend it out into colonies later. So this is how many human vaccinations or human medicines were developed with this cell line. Very, very important progress. No one in the world had been able to develop a cell line. And Mike did it on nights and weekends as a side project. And he's very, very good in the laboratory, but he got really lucky also. All right, another student who is defending his PhD Friday this coming up, which is kind of why I'm hurrying home. This is Matthew Smart, there with his dog, small photo. His project was huge. He collaborated with USDA and USGS in North Dakota. And the reason we're doing it in North Dakota is a long story I won't go into. But his question was, how does agricultural land use in North Dakota, where most of, 500,000 colonies are moved into North Dakota every summer just for honey production. And now North Dakota CRP is going out of favor and they're putting in more corn and soybeans. Honey production is going down in North Dakota. And we want to say, how does all of that agricultural land use in North Dakota affect the survival of honeybee colonies? And he worked with Zach Browning, a very good migratory commercial beekeeper. He has his bees in North Dakota. He has 20,000 colonies in North Dakota in the summer, and he moves them to California for almond pollination. So Matt chose 6 of his apiaries, Zach Browning's apiaries, and followed colonies individually for 3 years between North Dakota and California and back, recording their survivorship. And working with USGS, he could get land mapping of what was going on in a 2-mile radius around those colonies. And let me walk you through, this is an amazing study. So these color kind of graphs show you the 6 sites and what was in 2-mile radius, foraging range around them. And the more black or gray or white, the less bee-friendly it is. Okay, and I circled site 3 and site 6 for a reason, because site 6 was the best site overall, made the most honey, bees survived the best, I'll show you that, and site 3 was the worst surviving colony, and it's directly tied with what they had access to in their landscape. So he went through, not only having the landscape data, but he took measurements repeated measurements of all of these things. Colony level measurements, so what did the colony look like? Frames of bees, brood, how much pollen they collected. They had pollen traps on colonies, how much incoming pollen. We identified the pollen as far down to genus as we could. What pollens were they collecting? How much honey? Their parasites, their pathogens, their pesticide exposure. We took pollen, sent it in for residue analysis, everything. And then, not only that, but the most amazing thing was that he went into individual bees and did what I call, or what I had him call, a blood test. So you go into the doctor, they take some blood, they look at your cholesterol, and it tells you information about your nutrition, your genetics, and your survivorship, right? The risk of mortality, actually. So can we come up with a honeybee blood test? And what would we measure? And can we look at the nutritional status of bees? And does the blood test of an individual bee, of their blood, correspond to what they're getting in their landscape? And yes, it does. And these are just, you can't see a thing, but it shows you what different pollens they were collecting. And I'll tell you that at Site 6, that really good one, they brought in a whole lot of sweet clover, mellolotus. And I'll also tell you that mellolotus, sweet clover, is considered invasive, and they're trying to get rid of it in the upper Midwest. So one of the things I'm going to be trying to do with Matt's data is show the NRCS and the USDA and other interested parties that you can't support honeybee health at this time and get rid of mellolotus at the same time. Okay. This is the survivorship on average, all 3 years combined. This is how many colonies died yearly on average at Site 3, over 40%. And this is how many colonies died at Site 6, about 12% on average. Huge difference. And, and it was correlated with these measures inside the bees. So phytologinin is a storage protein in the blood. Very, very important for bees. This is the most important measure of their nutritional status and of how long the colonies will survive. Lipid stores, immune system activation. Those were the things that he measured that were highly correlated with the landscape. So now he's going to be moving to North Dakota for the next 3 years to replicate this study on a 5-state area to see how robust these are. Can we come up with a blood test so you can send in a sample of bees, run a blood test, and we can tell you about the landscape your bees are in. Okay. How am I doing with time? Nobody knows. Oh, I have 15 minutes. I'll probably get through this talk. That's good. Another student that will actually, she's been working for me, but she'll start her PhD degree this fall coming up. This is Morgan. And here's the question, and it spins right out of Matt's, is if we're going to get rid of sweet clover, or if there's any movement, what are we going to replace it with? Everybody says, let's go plant native plants. It will help our bees. But do native flowering plants, prairie plants in our area help honeybees? No one knows. Everybody says go plant them, and you do see honeybees on them. But can they make a honey crop from these? And how would they have to be planted in order for the bees to make a honey crop? So that's the background. They want to plant bee-friendly native prairie flowers and restore native biodiversity, which would be amazing for our native bees, but we have no idea if that would really help honeybees. So we know sweet clover and alfalfa are the major honey crops in our area, also basswood trees. But here's some alternatives. Are these honey-producing plants, if they're planted in high density? We have no idea. One over here. Which one? Mountain Mint? Purple Prairie Clover? Yeah. But you have to get it in really high density, I think. We don't have it like that. Anyway, so what she's done is go to some native restored prairies around the Twin Cities. Oops, I'm sorry. Around the Twin Cities, the ones with stars, are prairies that have been restored with really high numbers of native flowers, and not just grass prairies, but native flower prairies. And she puts colonies out there, puts pollen traps on them, and collects pollen every couple weeks, and she collects nectar, which, because all nectar has a little bit pollen in it, and we can identify the pollens from the nectar. And then we'll look at the pollens under a microscope, and I'm really interested to hear the talk later on in the afternoon how you're identifying pollens here. But this is what Morgan is doing, and she's just using microscopy to identify the pollens that the bees are collecting. So we don't know this yet. We do know that bees placed near prairies are flying and collecting sweet clover. We know that much, but we don't know yet how much of the proportion of their whole diet is these prairie plants. And this is a project we're in the middle of. Okay, I talked briefly about this project. Another graduate student, Ian Lane, who's working in urban environments, working on the bee lawn project, and trying different species to seed into an existing lawn or to start a lawn from scratch that's both grass and flowers that you can mow and maintain as a lawn, but they would continue to flower. And of course, this is not what you want to do for a soccer field or a baseball field or some of the front yards where your kids are actually playing. But this would be a landscape idea for areas that are just, in my mind, acres and acres. And in our area, fortunately, you don't have lawns so much here, but so many lawns that the only time anybody goes on it is when somebody's out there with a power mower. So there are a lot of water, a lot of gas mowing, a lot of herbicide, a lot of insecticide, and a lot of fertilizer to have a grass that doesn't even want to be there, because if you let it go, it turns into Creeping Charlie. So if our human aesthetic wants this manicured lawn, how could we at least make it a bee pasture or a bee lawn? So stay tuned on that one. He's about halfway through. He knows what kind of turf to use in our area. It's hard fescue, very sustainable, very low input turf. He's got a list of flowers that appear to withstand mowing, but we need to test it out a second year. Okay, another project just finishing up is Judy Wu. Judy Wu is married to Matt Smart, so she's now Judy Wu Smart. And it's nice to have a couple in the lab, and they just had a baby, and so like I'm a lab grandmother, which is really fun. So Judy studies pesticides, and in particular she studies the effects of the neonicotinoid insecticides on queen bees. So I'm going to show you the queen honeybee study, but she's also done one on queen bumblebees. Now these neonicotinoids are the systemic insecticides. They move through plant tissue, and they can get into the nectar and pollen of plants so that bees pick it up in their diet and take it home. In high doses, we know they can have acute toxicity that I talked about yesterday, where the bees die outright. And at very low doses, they can just get dizzy, disoriented, drunk-like, and maybe not find their way home. Okay, so that's kind of what they do. So one of the neonicotinoids is imidacloprid, is the compound name, and so she looked at its effect. So she kind of did a dose response at the colony level. We started with very small observation hives that were placed in these sheds that we have. So 1-frame observation hives and 2-frame observation hives. So 1-frame observation hive is just a very small colony that she fed, when you go back, imidacloprid, what does it say, twice a week in sugar syrup. Then she looked at 2-frame observation hives, and she looked at 5-frame nukes. The detailed measurements she needed to take, 5-frame nukes was as big a colony as we can get, but I believe that a 5-frame nuke is now a colony. It functions like a colony, more so than an observation hive. Okay, so she gave them 1 or 0 parts per billion, 10, 20, 50, and 100 parts per billion imidacloprid in 50% sugar solution twice a week. She started the colonies. She shook bees onto foundation, no comb. She let the bees draw out comb, and then, with the queen in there, she measured the queen's activity and the queen's egg-laying rate over 3 weeks. And after 3 weeks, now you know from a bee lifespan, they have eggs, larvae, pupae, and after 3 weeks, adults are starting to emerge. So she stopped the experiment at that point and collected residue in the workers and everything. And I'll show you a little bit of that data. So she's counting how many eggs the queen produces and how much brood they produce. And she tracked the queen's activity. So she put a transparency in front of the observation hive and 15 minutes at a time, several times a day, and she just tracked where the queens went, how active they were. That pink sheet is just behind the transparency so you can see what was written on it, right? And then after those 3 weeks, she pulled out the brood frames. So, of course, after 3 weeks, bees are starting to emerge here, right? This should be familiar with you, and here's brood that hasn't emerged yet. And she would look at the brood pattern, a 100-cell example of what the brood pattern looked like in those colonies. And there's lots and lots of data, but this one kind of says it all. So here's 0 ppb brood pattern, nice, and lots of it. Here's 10 ppb, 20, 50, and 100. So you can see the brood areas went way down, the egg laying rate went way down. She became spotty even at 10 ppb, which is a pretty sublethal level. And less brood and less activity of the queens. They spent a whole lot of time just hanging.
What would be representative of a field condition that would actually encounter?
Okay, so when you treat, so in our area, bees don't feed on corn pollen very much. We know that from all the pollen studies that Matt collected. Corn or soybean, maybe a little bit. But in those cases, when the seed is treated, what comes out in the corn pollen is about 4 to 5 parts per billion. If it's been planted corn on corn on corn, it can get up to 10 parts per billion. Flowering canola, same thing. If it's treated, seed-treated canola, the amount that you'll see in the nectar and the pollen of a canola ranges from 6 and up to 10 ppb. So those are pretty field relevant. The low dose, the 10 ppb, is field relevant, I would say. If you're in an urban environment and they run into nursery flowers or injected trees or anything that's in the city, you can get up to 20 to 50 ppb in flowers, no problem.
Marla, did you call sister queens then?
These were sister queens. So at the end of the experiment, now this shows the, okay, here's the legend. These are 0 ppb and higher doses, and it shows the total amount of brood, just some of the data in the controls, and you can see even 10, 20, 50, 100 ppb, less brood, total brood in that colony, and kind of all the same, just a little bit effectively. And this is the most important thing that's going on in neonic exposed bees, is that they don't bring back pollen. And if you look into a colony of bees that they've been exposed to, and this is shown here over and over, 1 frame, 2 frame, 5 frame, these are the controls, and here's all the other doses way down here. Controls bring in a lot of pollen and a little bit of exposure. We don't know if they get out there and they can't do it. Like, if they can't orient, can they not pack their pollen? So Judy right now is writing a big proposal to do postdoctoral work that I was reading last night to study this effect. Are they unable to physically maneuver the pollen, or do they not have an appetite for protein? Something's going on with protein in those colonies, and it's not, the other ones on here, it's not going on with nectar. They continue to bring in nectar into the colony, but not pollen. Yes, and I would say that yes, thank you. So much of the reason that they're not producing as much brood is they're not getting as much protein. Matt's research is showing that bees is critical that they have all this protein for survivorship. You can measure protein in their blood through vitellogenin. And this all comes back to what I was talking about yesterday with nutrition being so important for the bees. And the indirect effects of neonics may be nutritional, may not be toxicity to the bees directly. It may affect them enough where they can't bring in pollen which then doesn't kill the colony but has all these downstream effects where the colony just can't, it's not as healthy through its nutritional stores. So I'm going to end the talk and take questions now. Yeah.
How much of the stuff that you put the feed on, or the nursery, infected stuff? I mean, is that pretty common?
All right, so he wants to know how common it is for bees in cities. And let me start with bees in agricultural areas. All of the pesticide residue from Matt's study from North Dakota, we never detected any neonicotinoid, any compound for 3 years in North Dakota. None. Okay? In the cities, we haven't done all of those studies. Other people are showing them. But it turns out neonics are not the ones that are the most prevalent compounds that bees are picking up. This data shows if they do run into them, this is what happens. But all the big level field experiments, and not enough of them have been done in cities because they're used more prevalently in cities, it is probably not the main thing that they're running into, the bees are. So we know they have effects, but it's not the main kind of insecticide they're encountering with it.
Going back to your Propolis envelope, did you mention more or less chemical attachments to the sidewalls when you had the envelope built?
No, we used 9 frames in that 10-frame Langstroth equipment to make sure they had enough space, but they did not attach the combs, no. And over here?
Citrus growers in Florida and maybe in other parts of the country are trying to deal with citrus greening, which is infected apparently. All rows are using very intensive applications of neonics. And they say that because they're doing it below the surface, it goes directly into the plants, and they're doing it at a time when the bees are not feeding, that it's not affecting bees. How do you...
Yes, this is a great point. So citrus greening, and it's in Florida and it's also in Southern California. And they're using a lot of the neonics injecting citrus trees, or actually they're putting ground applications, heavy doses, big doses, into the ground that will run up the tree and into the citrus nectar. They're going to do a huge study in Southern California to look at that. And that is one example where, yes, the bees in a citrus orchard, where they're going for citrus honey, will encounter high doses. So, yeah, that study, it's about a 3-year study. It's being planned. It'll start fairly this season, I think. So stay tuned on that. But that is an example where, yes, the bees could encounter really high doses. And that's not a good thing. Yeah.
I read that when wax is tested, that they actually find more miticides in the wax than they do neonics and other pesticides.
Correct. So if you look at wax residue on brand new foundation or any colony, even if you haven't been using or never used Apistan or Coumaphos, it's in the wax and it's on your bees. Now another thing that Judy found that she plans to test also, this is super interesting, and we don't know if this is true, so I hesitate to throw it out there because I know what beekeepers do when I throw out a hypothesis. You think it's done in fact and everything, so having said that, she found in her little observation hives, in the colonies that were treated, they produced, she was feeding them sugar syrup, you have to in these little colonies. They were producing a lot of wax scales. Have you ever seen a bee have wax scales underneath its abdomen and how they come out? And then wax scales were falling to the bottom of the observation hive. So she collected them and sent them out for residue, and they were so full of pesticide residue. She was wondering if that's one way that bees get rid of pesticides. If you feed them a lot of sugar syrup, or they get into a good nectar flow, and they produce a lot of wax, can they shed wax scales to get rid of some of that pesticide? But if they have to use that wax to make comb, where's it all going to go? Right into the comb. So another very, very interesting question. And yes, your combs are contaminated. And sometimes at levels that are not problematic to bees, but it is out there, yeah.
If,
Last question, thanks.
if the neonicotinoids are not what they run into the most, you said, we see the results that they do. Do you know what it is? What are they running into?
The things that they run into the most are fungicides. They run into chlorpyrifos, which I think is an organophosphate used on soybeans. They run into pyrethroids, those classes of insecticides. Huge, huge amounts of fungicides. All right, on that note, horrible note, thank you very much.